Hello friends, good afternoon and welcome to the
Rh antigens are going to travel through the placenta into the mother's blood, and immediately, because we have now antibodies in mother's blood against the Rh plus, she is R negative, the mother's blood is going to start producing antibodies immediately, and these particular antibodies would attack the hostile antigens of the baby. So, that means the baby is constantly producing Rh plus and mother is producing the antibodies. If Rh mother had a previous Rh plus pregnancy which was too soon, or she has taken a transfusion with the Rh plus blood, then the antibodies against Rh are already present in her blood, and now with this present pregnancy some more Rh negative antibodies have started being produced. So, as a result of this cumulative effect, the antibodies from the mother would also through the placenta enter the child, and they would start destroying the blood cells of the fetus. This would mean that we have now erythroblastosis foetalis. So, that means the Rh negative mother and Rh positive father combination is a bit risky for the child, and therefore, it needs to be looked at in medical terms. The expectations that we had from Mendelian crosses till now was that all the individuals of a particular genotype must show a definite phenotype. That means, we have either this or, which means the two allelic forms. So, that means it is a matter of predisposition that all the seeds which have a genotype of capital R, small r must be round, and all the seeds with a homozygous recessive must be wrinkled. There is no question about it. But then, if we look at a field and all the individuals of a population, that this is what we expect, would be say heterozygous capital T small t, they must be tall and that too with the same height. Let us ask ourselves a question, does it really happen? If we go to a field and see, are all the plants exactly of the same height? Well, if this was to happen, then let us take an exception. The gene for polydactyly is dominant. Polydactyly means that a person would have more than 5 digits on the toes or on the hand or the fingers. So, that means, if this is a dominant gene, then all of us should have had more than 5 digits in our limbs, but then why is it that the majority of people have only 5 digits on each limb and not more? The gene is still present, yet all of us having 5 digits may give rise to children who have 6 digits. So, that means this comes as an exception to the Mendelian expectations. Let me ask you a few more questions. We know that lung cancer is one of the deadliest diseases. There are many people who have been found to have this gene for lung cancer, but in their life they actually never develop cancer, lung cancer. Now, why is this so? They have had a gene, why did it not function? Similarly, diabetes, which is so very prevalent now, we find that there are different individuals who are suffering from this metabolic disorder because there is not sufficient insulin. Then why is it that sometimes they do not suffer from this disease or this metabolic disorder at all, even though they have a gene? Now, this problem is a bit complicated because diabetes is a multifactorial disorder. So, that means what we are asking ourselves is, does the mere presence of a gene guarantee its expression? if the gene is there, is it a must that it has to show in all the cases this expression? Does the environment also have some role in modifying the phenotype, whether this gene would or would not function, and if at all it would function, when would it function in the lifespan and under what circumstances? So, that means these are some of the questions which are intriguing, which bring us to today's topic, and that is penetrance and expressivity. Now, these are two terms which are comparatively less understood and less interesting in genetics, because we are going according to straightforward stereotype Mendelian expectation, 3:1, everything is the same and there is no variation. Now, penetrance means that what is the ability of a gene or a gene combination to be expressed phenotypically to any degree. In other words, what we have to study is what is the frequency with which a particular genotype is going to manifest itself in the individuals of a population. Say for example, we have 1000 plants, and out of these particular plants, say 650 are capital T and small t. So, naturally we would expect that these particular plants with say another 100 capital T, making a total of 750 plants should be of exactly the same height. But then is it so? They must be varying. So, that means something is wrong as far as statistical regularity is concerned. So, that means this concept of penetrance refers to the statistical regularity with which a gene produces its effect when present in the requisite either homozygous or in a heterozygous state. We could have two types of penetrance. There is nothing much that we can explain as far as complete penetrance is concerned, because we know that complete penetrance means 100%, and 100% would mean as per Mendelian expectations, tall means tall of the same height, and a weight of a grain would mean exactly the same. So, that means all the homozygous dominant or heterozygous genotypes have to show identical phenotypes, and it is complete or 100%. So, that means all the homozygous recessive genotypes on the other hand would also be phenotypically identical. The examples of complete penetrance began with the popular arrangement of 7 gene pairs that Mendel chose for his classical experiments with garden pea. What we studied on the last turn, the alleles, namely A, B and O in the ABO system of blood transfusions, they also follow complete penetrance, and we would not see a person who is lesser O or a higher O. But you remember, we had something like a Bombay phenotype, and this was something else because there was another gene which was involved in the expression. Likewise, there are recessive alleles for the vestigial wings in Drosophila, and also the black coat character in the guinea pigs. This is also a dominant allele, and wherever there is a mating or a cross, the complete penetrance would be there. What intrigues and at the same time interests us is actually the incomplete penetrance, because it is here that the results are not according to what we expect. So, here what would happen is that the genes in the homozygous or in the heterozygous condition will not provide a complete 100% phenotypic expression, and then we will have to calculate how less, and the genotype would always be less than the expected figure of 100%. So, that means what we expect the phenotype would be, there would be a comparatively lesser percentage. Let us give some classical examples of incomplete penetrance. A while ago we mentioned about polydactyly, which means more than 5 digits in limbs. Now, we know that it is a dominant gene P which is responsible. So, Mendelian results would expect that capital P that is homozygous dominant as well as heterozygous, which is capital P and small p, should be polydactylous, and the normal condition would be that we have homozygous recessive. Now, in actual field experiments, out of the 84 people which were studied for the gene of polydactyly, they had this gene either capital P or capital P small p, that is homozygous dominant or heterozygous. Only 76 were found to be having actually a polydactylous condition. That means 76 out of 82. The penetrance is not complete. It is equivalent to 0.9, which is about 90%. Putting it in very simple terms, this gene has expressed itself 90% of its potential and not complete or 100%. Likewise, we have many other examples. There is another concept of brachydactyly, which means there are many people which we see around us have shortened and malformed fingers, and sometimes they are non-functional. So, that means the number of digits is 5, but then they are not working, they are disformed or non-functional. So, we find that this is only this character is 50 to 80% penetrance. What interests us the most is again diabetes mellitus, which people call as the royal disease. It is actually controlled by many genes, and we have seen that all the gene carriers amongst us may not develop diabetes. What is the different intensity, that we would discuss a bit later, but then they do not. Making a summary of complete as well as the incomplete penetrance, we find that the identical known genotypes would yield 100% of expected results and 100% expected phenotypes. There would be no exception at all. But in case of incomplete penetrance, identical known genotypes would yield always less than the expected phenotypes. There have to be some exceptions which would be not in accordance with the expected phenotypes. This brings us to another very important concept, and that is expressivity. Having 5 fingers or more than 5 fingers is one issue, but then there is so much of variation among these people themselves. For example, when we ask ourselves the question, why some polydactylous persons have fully functional extra digits? That means all these 6 digits are functioning as if a person has 6 functional fingers, others have just in the form of stubs and they are non-functional, they are just hanging and not taking part in, say for example, the grip of a person, or in case of a foot, they are not taking the weight of the person. So, that means this is a question which is of variation. And why do these extra fingers and toes occur in some person's left hand, in some person's right hand, in some person's foot, and so on. We have so many celebrities of the past and present. Hrithik Roshan, for example, has 6 digits in his right hand. The great cricket legend Vivian Richards was also polydactylous, or in fact is polydactylous. So, that means he has on the left hand, and this means that there is again a difference in expression of this penetrance. Penetrance having its existence is one thing, but then showing in different intensities, showing in different numbers is another. And therefore, if we have 4 people who are suffering from diabetes, all of them have different intensities of this disease. Somebody is taking one tablet, somebody is taking an insulin injection, somebody is taking an insulin injection twice a day. Why is it that all of them have the gene for diabetes, but the intensity of its expression is variable? All these variations are to be grouped under what we call as expressivity. So, that means penetrance and expressivity, these are two related yet distinct terms. Expressivity is basically the extent of expression of any gene. In other words, it would be the degree to which a penetrant character is expressed. In other words, the degree to which a penetrant gene or a genotype is phenotypically expressed in an individual. Now, it is here that we talk about the role of the environment. So, that means it is not only the genotype, it is also the environment which is going to perhaps modulate, which is going to modify the expression of the genes. So, in other words, when we are talking about the process of expressivity, we should remember that at the biochemical level, it would be the result of different degrees of function of the proteins which are being encoded by the gene. So, that means when a gene is translating into proteins, it would be the sheer kinds of proteins or the number of proteins which are produced, and above all after their modification, how are these proteins functioning, in what order, in what magnitude would decide how different types of diabetes patients having the same ailment are suffering differently. There are other examples of variations in expressivity. There is a disease called as osteogenesis. Now, this is a very costly disease because we find that the white part of the eyes, it becomes blue, and the person who is suffering from this also has a very fragile skeletal structure, having very frequent broken arms or fractures. The person can also be deaf. Now, these three ailments should be present in the same individual if one is suffering from osteogenesis. This is definitely an autosomal dominant disorder, and also it shows 100% penetrance. A person with this dominant gene has to have this ailment, but then the sufferer, it is seen clinically that the sufferer may have either these three ailments, all of them, or any two of them, or just one of them. So, that means no doubt this gene is 100% penetrant, no doubt it has a variable expressivity, but then the expressivity does not seem to be constant, it seems to be flickering. So, that means if one was to compare the constant expressivity as compared to variable expressivity, what we have just discussed about the osteo problem is variable expressivity. That means the identical known genotypes with an expressivity, they would affect the yield of range of phenotypes. There would be either all the complications or some of them. Whereas, if the expressivity was to be constant, then 100% phenotype would be expected of the Mendelian type. So, that means there is another very important aspect of incomplete penetrance, but variable expressivity. There is an ailment of the nerves which is neurofibromatosis. Now, this is basically an autosomal dominant disorder. The penetrance is to the tune of 50 to 80%, but it shows variable expressivity. People who are suffering from neurofibromatosis may have, say, very mild black or darkish blue areas on the skin. We all have some moles or some parts on our skin, but that is not neurofibromatosis. Neurofibromatosis is due to an autosomal dominant gene, and it is also fatal many a times, because in a very mild forms there is some distortion in pigmentation or depigmentation, sometimes even hyperpigmentation, but then under acute cases there are tumor like outgrowths, and this is not enough. The person suffering from neurofibromatosis would have high blood pressure, a large head, very frequent headaches, tumors in even eyes, in the brain and in the spinal cord. The whole spinal cord seems to be curved. So, that means these alleles are showing various expressions. Now, all these ailments that I have mentioned may all be together in one individual. Some of them may be in one individual and the others may be absent. So, that means whatever is the expressivity, it is also variable, and so is the penetrance here, because this gene is also penetrant to an incomplete manner. We find that the variable expressivity under very mild circumstances would mean that we have just these spots on the skin, but then large number of neurofibromas, which are tumor like outgrowths throughout the body, are seen, which make life quite difficult, and of course, there are the other metabolic disorders. So, that means if we have a variable expression of the similar genotypes, then okay, the genotypes are identical, but then they are in fact producing a broad range of phenotypes, because gene activation is variable, and on top of it, its variation is also variable. So, within a population, we have all sorts of different combinations, and this gene is also incompletely penetrant. If we were to look at the importance of environment, which is the modulating force of modifying the genes and affecting the final expression of the phenotype, there are so many parameters that the environment has to offer. We always talk of the common English dictum, nature and nurture, which means nature is genes and nurture is how an individual is being reared, under what circumstances of nutrition, of psychological pressures, and of course, of other environmental variables. The age of onset of different ailments or the action of genes is also variable, because we know there is always a programmed activation and deactivation of genes in our body. There are so many genes which express later in life. If you were to look at my head, I started balding when I was about 29. So, that means the pattern baldness appears in males between 20 to 30 years of age. There will be many like me who are also bald, and they must have started balding at approximately a similar age. But then the intensity of balding, the areas which went bald first, say from the rear or the middle or the front, they were extremely variable. So, this is also an example of expressivity. I would perhaps not be able to compare the pattern of my baldness with that to Prince Charles. He has a different types of balding pattern. Likewise, there is a Duchenne muscular dystrophy disease, and in this case the symptoms are going to appear in a child when he or she is about 2 to 5 years, and this would result in a progressive denaturation or degradation of the muscles. Likewise, we have seen in our families all around us that the disorder of diabetes also starts at somewhat a later stage, say 40 plus. So, that means this is an age related expression or activation. Having said that, I would also mention that there is higher incidence of diabetes in young people today. They have also a similar gene that their fathers and forefathers had, but then we find a much larger percentage of young people who are suffering from this disorder. Now, is it due to hypertension, their food habits, their lifestyle, and if I were to just to mention in a lighter vein a literary term, hurry, worry and curry, perhaps it is a combination of all the three which is driving this multifactorial disorder to express itself more. What do we mean that it expresses itself more? Now, the expressivity as well as penetrance percentage is increasing. The phenotypic expressions may not always be related to age, they may also be related to the sex of an individual, because we know we have an X and Y chromosome for a male and the two X chromosomes for a female. So, there are sex linked genes, and of course, the Y and the X chromosomal complements would have different expressions. There are different types of traits, say for example, sex limited, which means that they would be limited to a particular sex, either a male or to a female. For example, milk production in dairy cattle. This is again a matter of expressivity. Antlers and horns in males, the ability to produce sperms by the males and the ability to produce eggs by the females are also exclusive to either males and or to the females. Likewise, there are sex influenced traits, for example, pattern baldness, and this would also depend on the level of the hormone testosterone. And if we say that a character is sex influenced, then how does it occur in the members of the other sex also? So, for example, pattern baldness could also exist in a female, but then the probability is just 3:1. There are many people who have a cleft lip and palate, and here also this is a sex influenced character where the percentage in males is 3 times more as compared to the females. The distribution of facial hair in males is also highly variable, as you can see from the beard patterns of different males. The incidence of gout is 8 times more in males as compared to the females. On the other hand, the disturbing fact is that the ailment of rheumatoid arthritis, which is also a sex influenced gene, is 3 times more in a female as compared to a male. And also osteoporosis, which is due to the deficiency of calcium and vitamin D, D3. We have thrice as many females suffering from osteoporosis and a deficiency of calcium as compared to males of a comparable age. Similarly, there are many autoimmune diseases which can develop in our body all by themselves, and one of them being lupus erythematosus, where we have a disturbance with the RBCs, and here we find that the occurrence of this autoimmune disease in females is virtually 9 times more as compared to a male, and this would lead to all sorts of aplastic anemia and different types of anemic disorders in females. If we were to study other factors of the environment, temperature seems to be another very important factor which influences the expressivity. This is because many of the alleles of the enzyme coding genes are very sensitive to temperature changes. In other words, they are thermosensitive. A classical example is seen in case of Siamese cats and the Himalayan rabbits. They have a gene for tyrosinase, and tyrosinase is responsible for the synthesis of the pigment melanin. Now, when they are born, the young ones are absolutely milk white, and gradually their extremities, that is the tips of their nose, the tips of their ears and the tips of their tails, they are definitely exposed to somewhat higher temperature, and as a result the higher tyrosinase levels make more and more melanin, and their extremities they become dark. So, the Siamese cat and the Himalayan rabbit, they would change the pigmentation patterns. You can see that their ears, their toes and even their tail becomes dark, because that is being exposed to a higher temperature. So, that means these genes, they are expressing themselves somewhat more for the production of melanin subject to temperature. There are many chemicals also which are going to affect the expressivity of our genes. The most classical example which has been studied for a long time is that of phenylketonuria. Now, this phenylketonuria, which is commonly abbreviated as PKU, is the gene is located on chromosome number 12, and there is a defect in the biochemical pathway. Phenylalanine is synthesized in a very large scale. Ultimately, what happens is that the person's urine turns black. After some time, it gets oxidized. It is not just like that. There are so many disorders which ultimately would lead to a mental retardation at a very early age. Now, it is seen that the chemical phenylalanine is the culprit. It is the villain in the entire story, and somehow the whole story starts from mother's milk. When a child is taking the first gulp of mother's milk, then you would have a very large amount of phenylalanine getting into the child. The child starts making the different types of pathways. So, that means if one was to just locate a gene in a child and restrict mother's milk, even restrict the diet which does not have phenylalanine, we know of very few diets in the world which do not have phenylalanine, because all our cereals, all our milk products, they are all rich in phenylalanine. So, if one stops all these, then one can save oneself from this particular disease. This particular slide I particularly wanted to mention in a lighter vein, because there are two women in this photograph. They are actually, both of them are sisters. One of them, who is getting married, got diagnosed for PKU when she was only 3 days old, and then she was given a special diet. This particular diet was absolutely free from the chemicals which are, that is the phenylalanine, and she was perfectly normal in her physical as well as mental faculties. Her unfortunate sister, who is also shown sideways, who is basically the bridesmaid for this wedding, could not, and by the time she was diagnosed, the nutritional factors were perhaps too late, and she was suffering from mental retardation. So, that means we summarize our expressions of genetics, the genes and the environmental factors. What we come to a conclusion is that It is just the presence of a gene. Is it just enough to ensure that the phenotypic expression would come in the expected manner? Perhaps no. So, the phenotype of a particular individual or the expression of a particular gene would also be affected by what is the constitution of rest of the genome, because they are also going to influence the functioning of the gene per se. In other words, it is a whole complex of internal and external environmental conditions which are going to influence the activity of a particular gene, which brings us back to what we started with, nature and nurture. Sometimes it is nature versus nurture, and sometimes it is nature and nurture. A classical example of this behavioral difference would be that if we were to isolate two identical twins immediately after birth and expose them to different conditions altogether for say 20 years, and such clinical studies have been done, there would be many differences in their intellect, in their thinking, in their personality, and also in their metabolism. So, that means being genetically identical to the last core, because they are identical twins, it is the environment which would also influence the expression of those particular genes. Let me ask ourselves some very innocent questions, and then only we will be able to answer them or try to answer them in the next part of our lecture. We see people around us having different heights. All the advertisements of matrimonials say a lady is below average in height, above average of height, very tall. So, that means will all the children of a medium height couple always be medium in height? There will be no tall person in their family? And is it a must that the skin color of all the family members would be the same? Nobody would be fair, nobody would be a bit darker, and so on. This is a question which is very pertinent. What we see is, and we believe that all the Nobel laureates are very brainy people. So, if we look at the history, were their parents also that intelligent or equally intelligent? Putting it in an opposite manner, when these Nobel laureates became parents, is it a must that their children would be equally intelligent? In other words, are these characters of skin, height and intelligence, are they being perpetuated into the next generation in toto? Is it a guarantee that a tall person would always give rise to or give birth to tall children? Now, this seems to be a very old and decadent idea of Lamarckism, which means the son of a blacksmith would be as strong. No. So, that means the simple idea is that if you give a mathematical problem to all the members of a family, would they be able to solve it with the same finesse, with the same intellect? Perhaps not. There are other questions also which we can ask ourselves, and that is, if we look at big hectare of field, are you sure that all the plants are there can be really categorized into tall and short? Are there not any intermediate heights of plants like our heights? And why do we always say that a person is short, medium, above average, and so on? So, that means if we were to look in the market for different fruits and vegetables, why is it that there is one apricot which is larger than the other? The tomatoes, they are different in their shapes, in their sizes, and of course, in their colors. Why is it that the different babies, we say, oh, it's a bonny baby, it was a very heavy one, and the other one is too feeble and too light? So, that means why are they born with different weights? So, that means now the time has come to think beyond the two alleles, beyond the two genes, because till now we have always followed a dictum, this gene is represented as this or that. By saying this or that, we mean that there are only two alternatives. Now, we should start thinking this, that and that too, which means there is some other alternative other than just two of them. So, that means this would mean that there are certain intermediate characters also. To give you a few illustrative examples, the beautiful flower of Gaillardia pulchella would show quantitative variations, which means that their petals have different colors, their diameter is different, the number of floral pods in terms of calyx and corolla, they are also different. All of them look beautiful, but then they are different. Their genotype is identical. Likewise, the bracts, which are petaloid or colorful in the Indian Paintbrush Castilleja hispida, they show just two extremes of color range, which means red and yellow. But then actually you would see plants which have a whole lot of phenotypic range. Some of them are orangish, some of them are yellow. So, that means the bracts are variously colored. Here you cannot simply say that we are talking of red and yellow. There are red, there are yellow, and there are some intermediate variation. Now, how does Mendelism account for these variations? And the simplest observation is when you go to the market, there are tomatoes of different sizes, different colors, different shapes. All of them are tomatoes, all of them seem to have the same genotype. How do we explain all these questions that we have asked ourselves? We would try to explain them on the basis of quantitative genetics. That means we are going to add things together when we are going to talk about the intensity, lesser or the larger intensity. So, these are very small differences which were actually observed by people in the past also, but then there was no sound scientific explanation which was there. People just call them as continuous variations. Okay, these are variations, they go on and they are perhaps arising as a result of environmental changes. Some potatoes are large, the others are small, and they could not store that much of starch. But why? We could not have any sound answer. And it was not hard and fast that a larger fruit would always give rise to a plant which would later have larger fruits. It was the German scientist called Reuter who was the first person who worked on tobacco plant Nicotiana, and inadvertently he saw certain results which are the basis of today's quantitative genetics, although he did not know that how important work he had done. He only said that there are different sizes of plants and this seems to be a continuous variation, right. And should we be done? 1 minute. Okay. We have the history of quantitative genetics on us, and it would be in fitness of things to talk about the quantitative genetics in details on the next turn, right?
Right. Thank you so much, sir. In fact, it was an extremely interesting lecture that we had today, even for the general viewer who's perhaps not a student of botany, to learn the idea that perhaps it is not just about the genes, there is much more to it, and we got to know through various examples through you. So, extremely interesting and grasping and very involving. So, I would also request the viewers to perhaps even go back and watch the lecture again, as I am going to do. And with this, we again thank sir for being here today. Sir, thank you so much for your valuable input, and we expect sir to see here more on future occasions and talk more about ideas.
And there are many more exciting things in store.
Oh, that's great, sir. Wonderful. And friends, thank you so much for watching. Have a wonderful evening.
Thank you.